Hello, everyone, and welcome to today's webinar, Identifying Cellular Diversity Through Single-Cell Genomics. I am Michelle Ashton of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Beckman Coulter. To learn more about our sponsor, please visit www.beckman.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Abbas Rizvi, molecular neuroscientist, Zuckerman Mind-Brain Behavior Institute at Columbia University. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Abbas, you may now begin your presentation.
Thank you, Michelle. I'm honored for the opportunity to present, and thank you all for joining. The goal of the presentation that you will see is to understand, by way of highlighting examples, how single-cell sequencing may be utilized to understand the establishment, development, and maintenance of cellular structures. I'd like to begin just by simply highlighting the following example. Imagine if you had a group or a clonal population of cells on the left that are all identical, and over time, these cells differentiate into a heterogeneous population of cells that you see on the right. This example is an embryoid body, and these mouse embryonic stem cells are transitioning into motor neurons. But of course, this is just one example of where a group of cells that are highly similar can suddenly become very different from one another. And this has unique importance when you think about a system like cancer, where you want to understand what's happening in a tumor as it progresses and how are cells within a hypoxic center different from those at the margins of that tumor. So clearly what that example highlights is that understanding individuality is of paramount importance, both from a basic science perspective, but also in consideration of translational importance. So how do we gain access to cellular diversity? How do we understand how one set of cells, which are seemingly identical to one another, can become so unique and different? Well, if we highlight the following example. If we take a complex tissue type like the spinal cord within the central nervous system, and I'll have more to say about that in a moment, and we were to isolate individual cells, for example, by using flow cytometry, or alternatively density centrifugation when you're looking for nuclei, can we generate cDNA from single cells, sequence and map against the transcriptome, and then analyze in a way where we can find unique markers for each particular cell type? And in an exciting direction that will be the subject of possibly a different webinar, we could then multiplex in situ sequence to be able to understand where those cells are spatially distributed within that complex tissue. So what we'd like to be able to do is go from a complex tissue and understand all of these different cell types throughout development and an adult, and be able to create atlases of these cell types. And when we want to do something like that, we really want to consider the layered and stratified nature of gene regulation. When we consider gene regulation, we understand that genes have a particular structure associated with their regulation, and that's dictated by promoter sequences near the transcription start site where the basal transcriptional machinery is capable of binding, and that there exists enhancer elements, both proximal and distal, which bind transcription factors, and through action at a distance, we have stimulated transcription and subsequent splicing. But we also have to consider that there are other factors. For example, we know in prokaryotes that there exists transcriptional repressors. We don't have that in mammalian systems. Rather, we have other mechanisms at play. And the example here is if we consider chromatin architecture, and we understand that closed chromatin is refractory to gene expression, whereas open regions of chromatin enable activated gene expression. Well, you could either look at the system by considering transcription, or you can think about chromatin architecture when trying to understand cellular diversity. And what that really speaks to is that there are multiple experimental readouts of cellular diversity. I just told you about DNA accessibility as one example, but you could also have long-range chromatin interactions. So for example, someone can do single-cell Hi-C experiments to be able to understand cellular diversity. And of course, when we look at activated transcription, we're looking at RNA sequencing. And then finally, we could try to understand something about protein abundance in individual cells and try and gain some sense of which cell is which. For the purposes of our conversation today, we'll focus on single-cell RNA sequencing, but I do want you to be aware that there exist workflows that enable you to look at such systems in a multimodal fashion. That is, by combining two or more of these readouts with one another. But let's get back to the spinal cord. Everybody here can relate to the following scenario. There's a flame. There's an individual who is burned by the flame, and there are pain receptors in the skin that then take those sensory afferents and deliver them to the spinal cord and up to the brain, which then sends motor commands out down to through the spinal cord, and that you have motor afferents or outputs, which are mediated by motor neurons that innervate muscle. Ouch, that hurts. Your hand retracts. And so what somebody might want to be able to do, me in particular, is trying to understand all of the different cell types that would mediate our ability to respond, for example, to experiencing pain and subsequently being able to take action. But this is actually a pretty complicated set of circumstances. When we look at the human spinal cord, we know that there are 31 segments along the rostrocaudal axis, and that is from cervical down to sacral. And we understand that cellular diversity is largely a function of where you are along that rostrocaudal axis, whether you are in the cervical, thoracic, lumbar, or sacral regions of the human spinal cord. Furthermore, we understand that in the middle, if we look at a cross-section of the human spinal cord, we see that it's an incredibly complex structure whereby there exists sensory neurons that largely occupy where afferent sensory signals come in, and that we have motor neurons down towards the bottom at the ventral horn, which then would send motor output. And when we take a more granular look at the spinal cord, we see that there exists a Sargasso Sea of cell types that are patterned in this rostrocaudal and dorsoventral manner. Now, of course, we know that there exist neurons, both excitatory and inhibitory. There exist astrocytes, which are the support cells for these neurons. Microglia are the immunological guardians of the central nervous system. There exists oligodendrocytes, which form myelin sheaths that protect the axon. There are also ependymal cells, which provide a critical barrier between the cerebrospinal fluid and the remainder of the central nervous system. Now, when we think about such a complex situation and trying to understand how you can develop and maintain such a complex structure, we want to try to reduce the system down into something that we can understand one cell at a time. And so what I'm going to discuss with you is the possibility of understanding neurogenic commitment of motor neurons with single-cell resolution. Now, of course, all cellular diversity begins at fertilization, whereby you have an egg that upon that fertilization subsequently divides into a blastomere, as you'll see on the right, and then through subsequent development, you get a beautiful central nervous system, as you'll see in this embryo, that is formed. And in green is the spinal cord labeled in this clarified embryo. And one thing that we can do with these blastocysts is we can isolate mouse embryonic stem cells, and we can actually differentiate these mouse embryonic stem cells into a wealth of different neuronal cell types. And so what we'll discuss in a moment is the directed differentiation of mouse embryonic stem cells into motor neurons. And that provides a platform to be able, in vitro, to understand how motor neurons arise from a clonal population of cells. So let's go back to the example that we started out with at the beginning of this presentation. Imagine that I have a collection of mouse embryonic stem cells and that I'm looking at them over time over a 6-day differentiation protocol, whereby I induce these mouse embryonic stem cells twice, on day 2 with retinoic acid and sonic hedgehog, and on day 4 with retinoic acid and sonic hedgehog. Now, these mouse embryonic stem cells are actually special because they contain a transgene for motor neurons, that is HB9, driving the expression of EGFP. That means that all of the green cells that you see on day 6 are, in fact, motor neurons. And so the question is, how do these cells actually transform from embryonic stem cells into the diverse population that you see on day 6? And in fact, this is of critical importance because when we're trying to understand the biology of motor neurons, we can actually dissociate these green cells, sort them using flow cytometry, and be able to culture them and subsequently study disease states. So of course, hopefully this motivates you why it's important that we understand how this diversity comes about over time. But there are some considerations that we need to really take into account when we're trying to understand a population of differentiating cells. Imagine the following thought experiment. I begin with a clonal population of stem cells at time point 1. And they're all highly similar to one another. And when I look at the middle boxes, in this case, you see all of these cells, if I were to profile their transcriptome, because the cells are all similar to one another, if I took all of the cells and profiled them in bulk, or taking the ensemble average, that ensemble average would reflect what's happening in this dynamic system. But if I look at time point 2, you would see in the differentiation trajectory that some of the cells are a little bit behind and show some stem cell characteristic, but at a branch point, they start looking more like cell type 1, and another population is differentiating towards cell type 2. Now, if I took the ensemble average of that transcriptome over that entire population, it would belie what's actually happening on the right with single cell resolution. And of course, when we look at time point 3, we now have fully differentiated cell type 1 and cell type 2. And of course, doing bulk RNA sequencing would certainly not give you any felt sense of what's actually happening. But now, if we did single-cell RNA sequencing, we may be able, if we're clever enough, to be able to contend with the asynchrony branched nature of differentiating processes. So let's think about an experimental design. Remember, I told you that we have day 2 through day 6 of our differentiation ongoing. And if we were to take these individual embryonic bodies and we were to dissociate them and sort into individual wells of, say, a 96 well plate, or it could have been a 384 well plate, We can utilize SPLIT-Seq, which is a published protocol, but we've optimized using liquid handling systems to be able to reverse transcribe with an inline cell-specific barcode in each well. We can pool the cDNA resulting from the reverse transcription and in vitro transcribe and generate a cDNA library for sequencing. And we did that over 2,743 single cells, yielding 7,000 genes detected per cell. But what do you do when you actually have data like this? It's actually not exactly trivial. How do you contend with trying to analyze and interpret results that may come through from an experiment like this? And the idea that I had was thinking about considerations of shape. Now, I had read a publication that gave some information about topological data analysis as a method to be able to understand how shape might help you gain access to biology. So let's take a quick break from single-cell sequencing and think about topology. Topology is all about understanding invariant shape associated with any three-dimensional structure. So by way of example, there are mathematicians that sit and think very carefully about a double toroid system like we're looking at on the left. And what they seek to be able to do is create reduced representations, that is to say, a skeleton. And that skeleton actually tells you something about that shape. So when we look to the right, this is known as a Reeb graph, and that tells you that, gosh, I have actually two invariant holes if I went from the top to the bottom of that particular structure. And that tells you something about shape. But of course, the real world is not continuous structures. In fact, our measurements are discrete. And so we positioned ourselves to be able to utilize topological data analysis. And in topological data analysis, what you're doing is you're taking a collection of data points, as you would see on the right, and inferring that they actually are a circle, as you see on the left. And of course, that gets increasingly difficult as your data becomes more complex, even in the bottom case where you're looking at two circles. So how do we actually do topological data analysis when we're thinking about high-dimensional single-cell data? So when you generate single-cell RNA sequencing data, one might think that each data point could potentially be a 24 by 1 vector. And so that constitutes a very high-dimensional data set when you're considering a large number of individual cells. And those data reside in a high-dimensional plane. And so on the left, we call this point cloud data. And as with any kind of high-dimensional data, we dimensionally reduce as represented by F1. Now, the F1 could be principal component analysis. It could be T-stochastic neighbor embedding. And you dimensionally reduce your data into a lower dimension embedding, as you see along this real line, as I show you right in the middle of the slide. And what we can do is we can create patches along our dimensionally reduced data. And with those patches, we can then refer back to the high-dimensional data that you see on the left, and we ask how are those cells actually related relative to one another in the high-dimensional plane. And once we've done that, we can actually then create nodes. So for example, in green, we see a set of cells that have a highly shared transcriptional profile, and we can group those cells into a node. And we see in red there are two other nodes of cells that actually have highly shared transcriptional profiles. But you notice that between the group of cells in red and the group of cells in green, that there exist cells that have shared transcriptional profiles. And those actually fall into edges of these graphs. And so what this allows us to potentially do is to be able to capture continuous structure existing within single-cell data. So let's just review. This is the point of the webinar, and trust me, I've seen this talk before, where everybody should just kind of sit up, put your phone down. If you're surfing on the internet, maybe stop for just a moment. A node represents a microstate, a group of cells that have a similar transcriptional profile. An edge between two nodes represents a shared group of cells. So let's make sure we're all on the same page, because we'll begin thinking about topological representations of single-cell data quite extensively moving forward. So what we did was we took our data from our single-cell differentiation, and we then created a topological representation. And what we see, and it's actually quite striking, is topological data analysis perfectly ordered these differentiating cells. On the left, you see mouse embryonic stem cells from day 2, rapidly transitioning into neural precursors, which you see in a lighter blue, and then multiple paths that bring you to progenitors, and then subsequently motor neurons, which are colored in red. And of course, that occurs on day 6. Now, of course, as an experimentalist, you want to be able to look at your data and sanity check and say, well, is this consistent with biological understanding that is known about this system? And so what we can do is simply highlight and ask, if I have known markers for each of these given cellular states, where do they reside within this topological representation? For example, if I look at OCT34, which is a Yamanaka factor, it seems to be highly represented in mouse embryonic stem cells and dying down as we move towards neural precursors, and you can see that in light pink. OLIG2 labels progenitor cells that give rise to motor neurons, and VOX is an acetylcholine transferase that actually labels motor neurons. So everything seems to jive with expectation, and that's good. However, what we want to be able to do is build out a statistical method to be able to analyze and interpret these data without using any kind of bias. That is to say, without anchoring ourselves in knowns about this system, can we actually use this as a platform for discovery? So we set out to create that statistical framework by first trying to infer a timescale for progression. Usually in sequencing experiments, we want to think about pseudotime. That is, creating a transformation that instead of thinking about experimental time, we're now parameterizing in context of transcriptional state. And so here we did that by defining a root node. That is a node that maximizes the correlation between graph distance and chronological sampling time. And in doing so, we're actually able to transform ourselves into pseudotime. But in addition to that, what we also want to do is we want to get some sense of confidence about where genes are expressed and localized along these types of graphical representations. That is to say that I want some sense of a p-value, that I have some confidence in my results. And so what we then define is gene connectivity, and that quantifies if nearby cells in the graph express the same gene transcript more than random. So on the left, you see VOX, which we would expect to see actually highly localized in motor neurons, and of course it has an extraordinarily low p-value. Whereas if we take a gene that is expressed scattered throughout the graph, we then see that its p-value is subsequently quite high. But we also want to know where we are along that graph. And so what we did was we utilized two metrics, centroid and dispersion characteristics, that tell us where are transcripts actually appearing along these graphs and how do they distribute themselves across that. And when we actually take the centroid and dispersion relationships in these graphs, what we're able to do is, in an unsupervised way, be able to identify cellular states, that is to say, sets of transcripts that are uniquely expressed along these topological representations. Now, biologically, why is this important, and what can we potentially learn? When we take centroid and dispersion relationships, what we're capable of doing is, in an unsupervised way, being able to identify cellular states associated with this dynamically differentiating system. And that's important biologically, because we know that there exists coordinated transcriptional regulation associated with cellular differentiation and development. And this allows us to see, as you can see on the left, we can find coordinated gene expression that occurs between pluripotent cells, neural precursors, progenitors, and neurons. And as you can see on the left, you can actually see waves of transcription associated with neural development. And in fact, what this allows us to do in an unsupervised way is be able to identify new markers associated with cellular development. So, for example, we find PCAM1 as a unique marker of mouse embryonic stem cells, and we can actually use immunohistochemistry to be able to validate these results. So we see on our topological representation that we can call out PCAM1 as a novel marker of mouse embryonic stem cells, and in fact we can now stain embryoid bodies against PCAM1, OCT3/4, and on the very right, looking for HB9 GFP, or MNX1 is another name for it, GFP expression. And what we can see is a perfect co-localization between PCAM1 and OCT3/4 in these mouse embryonic stem cells. However, when we look at day 5, what we're able to see is that in addition to OLIG2, we can discover that the endothelin receptor B also uniquely labels our progenitor population. And in fact, our immunohistochemistry in an individual embryoid body shows perfect co-localization. Interestingly, we also find that SLC10A4, which is a bile acid transporter, is a novel marker for motor neurons. Now, this is the kind of data that you find in single-cell sequencing, especially when you're doing an unsupervised analysis. Why would a bile transporter be appearing in motor neuron population? And would that be consistent with what you would find in vivo? And when we look at an embryonic stage animal, looking at SLC10A4, this bile acid transporter, we find absolute congruence between HB9 GFP, that's the marker for motor neurons, and the expression of this bile acid transporter. And you can see the motor neuron pool, and you can see the two limb innervating axons coming out, and they're perfectly stained with an antibody against SLC10A4. Moreover, what this type of analysis allows us to do is to be able to discover expanded transcriptional repertoires associated in a functionalized manner. That is, by looking at gene ontology, we can now look at regulators that are now associated with the transition from pluripotent cells to neural precursors, motor neuron progenitors, and neurons. Now you can see why this, of course, is of paramount importance and why this could actually lend to a generalized workflow that is of sufficient power and generality that can be applied to any differentiating population. And I argue that because it gives you the opportunity to be able to identify key transcriptional events and pivotal regulators associated with any type of cellular transformation. And of course, we've applied this workflow to other published data sets, and what that has allowed us to do is even identify hidden populations of single cells associated with a differentiating population. Here we're looking at pre-implantation embryos, and we can already begin to determine subpopulations of the trophoectoderm that were not previously understood. But let's go back to perhaps the more complicated set of circumstances. What if we wanted to be able to try to look within the human spinal cord and not just focus on motor neurons, but try to understand the repertoire of all cell types that exist within this complex tissue? Well, we initiated our work in trying to build an atlas of the human spinal cord by utilizing SPLIT-Seq. It's a mechanism of combinatorial indexing developed by the SELIG lab at the University of Washington. And in SPLIT-Seq, what you're doing is first reverse transcribing with the first barcode, but now you don't have individual cells in each well of a plate, but a collection of cells. And each of those collections of cells receives a first-round barcode. You then pool those cells, redistribute them to a new plate, and you ligate a second barcode onto your cDNA. You then pool and split again and ligate a third barcode. You can then redistribute these cells. You can lyse them in PCR with a fourth barcode. It's a probabilistic barcoding scheme that allows each cell to probabilistically receive a different first, second, third, and fourth round barcode. The details are well worked out as to how this occurs in a molecular scale. You begin with a round one barcoded primer, and then, of course, as I mentioned before, then at the 5' end, begin ligating second round barcodes block, then apply a third round barcode and a fourth round barcode at the very end of this workflow. But of course, one could imagine, sure, you could do this by hand, but that could be somewhat difficult. But we've managed to be able to get this to work at a pretty high level by automating using liquid handling. And so here you take stock plates of nuclei and distribute them along your plate, and then reverse transcribe, and then begin pooling and splitting in an automated way for each barcoded round. And what we're able to do, and this is an example over a large number, tens of thousands of single cells, in fact, nuclei isolated from the human spinal cord, specifically the lumbar region, we are able to identify ensembles of astrocytes, neurons, oligodendrocytes, microglia, so on and so forth, along topological representations. And so hopefully what this motivates for everyone participating in this conversation is that topological data analysis provides a meaningful path to characterize continuous transcriptional trajectories in single-cell developmental studies as well in atlas studies. And certainly integrated workflows where you can combine flow cytometry and liquid handling really enables unfettered access to cellular identity and the construction of cell atlases of complex human tissue. Finally, I'd like to acknowledge the team that I work with to be able to accomplish my work, and of course, the generous support of viewers like you. Thank you.
Thank you, Abbas, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of the presentation window labeled Ask a Question and click on the Send button. Abbas will answer as many questions as time permits. Okay, let's get started. Our first question is, you haven't discussed droplet-based approaches. Is that a method you just favor?
Certainly not. I mean, we also utilize droplet-based approaches in lab. However, many of the things that I've discussed with you here are things that are not particularly off the shelf. They are things that allow us to tweak and optimize, and certainly that can be done in a droplet-based format. However, we find that we get a little bit more flexibility when we're using flow and liquid handling.
Thank you. You've made a pretty convincing argument that flow cytometry can offer value in single-cell studies. Is there a reason to believe that flow cytometry can alter a cell's transcriptome and skew your results when you build your atlas?
Yeah, that's actually a really good point, Michelle, and thank you for asking. Here in our atlas studies, we're actually isolating individual nuclei. So these nuclei are isolated from post-mortem human tissue. And there we don't see any kind of changes, of course, because the nuclei are no longer actively transcribing. However, there are ways with flow. So let's say, for example, someone wanted to study something in a mouse where they had fresh tissue from a human. What you'd want to be able to do is dissociate for individual cells, but there are treatments like utilizing actinomycin that can actually block active transcription. And so there are many ways to be able to contend with the possibility that flow may skew results. in our experience, if you are doing this just right, usually everything will work out where you'll actually get something close to a transcriptional state that is comparable to what's resident within tissue.
Thank you. Some of the analysis you've shown seems somewhat advanced. Are there analytical strategies that can help newcomers to the field interpret their data?
Yeah, so that's actually a really good question, and usually that tends to be the barrier to doing any kind of single-cell genomic work. I will say that there are plenty of tools that are available. Seurat is one example. You have ScanPy, which is a Python-based implementation of single-cell analysis tools that are quite accessible. And so, you know, one of the wonderful things about single-cell sequencing is that it really has rapidly evolved, and it's through the good work of computational biologists that there are tools like that that enable people right away to be able to analyze and interpret their data. That having been said, I always recommend that a computational biologist be associated with kind of a first pass of trying this, just to make sure that you're doing everything correctly. But there absolutely are plenty of tools available. In fact, the analytical approach that I mentioned here with topological data analysis, we have available on GitHub. It's called SCTDA, and we have an entire software suite that enables, in a very open source way, anyone to be able to build these kinds of topological representations and statistically analyze along these graphical representations. So yes, there's absolutely tools out there, and they are quite accessible.
I would like to once again thank you, Abbas, for your time today. Do you have any final comments for our audience before we go?
Yeah, I think that, again, although I focused on neuronal systems or systems within the central nervous system, I really think that it's important to recognize the power and generality associated with single-cell genomics and the biological insights that can be afforded by doing these types of experiments. And so if you are hesitant in any way to give these things a shot, consider really the massive benefit of looking at cellular systems one cell at a time.
Thank you. I'd like to thank the audience for joining us today and for their questions. Any questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank Labroots and our sponsor, Beckman Coulter, for underwriting today's educational webcast. This webcast can be viewed on demand. Labroots will alert you when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's event. Until next time, goodbye.